Assalamualaikum warahmatullahi wabarakatuh to the evaluators. My name is NURUL HUSNA binti ISMAIL. My matric number is 1171476. My supervisor is Dr. Latifah binti Karim. Today I would like to present to you my proposal topic, which is pretreatment and enzymatic saccharification of durian, Durio Zibethinus seed for sugar production. The outline of my presentation, including introduction, problem statements, literature review, materials and methods, expected outcome, Gantt chart and references. For introduction, according to Jeropon, ripe durian seeds contain 51.1% water, 46.2% carbohydrates, 2.5% protein, and 0.2% fat. Recent studies led by Li and Chen stated that the seeds can be converted into sugar and various products such as flour, gum, emulsifier, and as food stabilizer. Lignocellulosic biomass consists of cellulose, hemicellulose, and lignin, stated by Zub, Lamy, and Pais. There are three problem statements in this research. First, abundance of agricultural biomass waste of durian seeds can lead to environmental issue. Second, limitation of sugarcane plantation affected domestic sugar industry. Third, high calorie content of table sugar. The purpose of this research is to evaluate potential of durian seed as alternative source of sugar production. There are three objectives throughout this research. First, to investigate the effect of pretreatment and enzymatic saccharification process on durian seed for sugar production. Second, to determine sugar content in durian seed using Benedict test, refractometer, and blood glucose meter. Third, to evaluate calorie content of sugar from durian seed using bomb calorimeter. Next, for literature review, a research conducted by Obeng and co-researcher used two types of durian pills, which are motong and chani, in pretreatment of phosphoric acid. The results shown the highest hydrolysis and glucose concentration obtained after 72 hours from 75% concentration of phosphoric acid in motong and chani pills. The higher glucan was observed in motong pill rather than in chani pill. It can be concluded that phosphoric acid pretreatment caused enzymatic hydrolysis cycle increase in glucose concentration. Previous studies by D. Muhammad Nazrul Hisham shown the presence of phytosterol in durian seed, which can be decreased atherosclerosis and heart disease. Research about pineapple waste included core, peel, and crown led by Roja and co-researchers. The results obtained shown pineapple core had the highest value of fructose, glucose, and sucrose, and the sucrose is a major sugar present in pineapple waste. A study led by Akara Chanayakon and co-researcher used banana peels cultivar to evaluate effect of banana variety on amount of reducing sugar, and the results obtained was Cloy Homtong produced the highest reducing sugar, while Cloy Kai peel had the lowest amount of reducing sugar. The optimal condition for the maximum reducing sugar yield were alpha amylase at 86 degrees Celsius for 75 minutes and glucoamylase for 60 degrees Celsius for 75 minutes respectively. For materials, it divided into two parts. Firstly, raw materials, where durian will be obtained from the local night market in Alostar, Kedah. Second, chemical and reagent will be used in this experiment, which are hydrochloric acid, sodium hydroxide, commercial cellulose, and others. First of all, sample preparation is the first method among the others, where durian seeds will be washed, cut, oven-dried, mashed using mortar, and then it will be sealed in vacuum plastic bags before being stored in incubator. Second is pretreatment, which is to weaken structure lignocellulosic component to make it accessible to enzyme and chemical for further processing. For untreated, 1 gram of durian seeds will be prepared for enzymatic hydrolysis with the undergo pretreatment process. For acid and alkali pretreatment, there are two parameters will be used. First, different concentration, which are from 2% up to 10% for both hydrochloric acid and sodium hydroxide. Second, different temperature, which are 30, 60, and 90 degrees Celsius for both acid and alkali. The steps for two parameters for acid and alkali pretreatment are same, but different at parameter used. Third is enzyme saccharification, where the cellulose converted into glucose. In this process, enzyme cellulase citrate buffer tetracycline will be used, then will be conducted in shaking incubator. The supernatant will be subjected to sugar analysis method. In order to determine sugar concentration, There are three different types of process will be used. First is Benedict test to identify reducing sugar in Durian seed. The glucose content will be mixed with Benedict solution, and then the color changes of sample will be recorded. Refractometer by dropping 2 to 3 drops on the present surface, and the reading of sucrose will be measured in Briggs unit. Blood glucose meter, dropping sample solution on tip of blood glucose meter strip, and the glucose content will be recorded in molarity unit. Calorie content of glucose from durian seed will be determined using bomb calorimeter. The sample will be mixed with flour to form pellet and will be filled in calorimeter, then the heat content of the sample will be determined. Statistical analysis by using Minitab, one-way variance ANOVA will be assumed significantly different if p-value below than 0.05, and the value will be expressed as mean plus minus standard deviation with triplicate independent analysis. Expected outcome. It is expected that sugar will be produced from durian seeds through pretreatment and enzymatic saccharification method. Sugar content is predicted to be measured using different methods, which are Benedict test, refractometer, and blood glucose meter. Calorie content of sugar from durian seed is expected to be lower than table sugar. The right picture shows white liquid sugar form that will be produced at the end of this experiment, while the left picture is white sugar, which is a regular form in sugar industry. This is my Gantt chart where my target completion of the research is on December 2020, and I will start my final writing on thesis until thesis submission on February 2021. The laboratory experiment will be held from September until December 2020. Okay, these are my references. That's all from me. Thank you for your attention on my proposal presentation.